as if these systems have functional value that have put them in an environment where they can succeed. And so one of my goals as a teacher is to train the student, allow the student to have enough time practicing using the science process that they can begin to develop their own hypotheses, their own strong hypotheses, and start to speculate scientifically. Why is it, for example, the developing of a lot of good questions? Why is it that the mackerel does not have a swim bladder? What about the mackerel's design allows it to survive without this anatomical feature that almost all other bony fish have? What is it about the type of symmetry of the shark's tail that allows it to more capably swim, to more capably be a predator in a vast oceanic ecosystem? And so looking at anatomical features in particular and being able to ask those questions. An evolutionary paradigm often shuts those types of questions down with reference to the distant geological past. Junk DNA is a very notable example. All of this extra DNA that doesn't seem to code for actual proteins. It must be junk that's left over from eons of time, as we have seen common descent occur from a single-celled ancestor billions of years ago. Another example would be vestigial organs. Rather than saying, well, this organ is non-functional, that means it probably is left over from the divergence of birds from the rest of the reptile line. Rather than assuming non-function, why don't we study the design of the creature and determine what its function is? And so this is something that's very, very plain and simple and easy to do in the biology classroom, especially with dissections. Dissections are such an important part of this. I like to use fresh specimens for this reason as well. These creatures have been designed a certain way. Let's experience them the way they've been designed. Let's smell that incredibly strong oily fish smell. Let's feel the mucus that's coating their scales. Let's see the heart that's actually crimson red and its tiny two chambers. Let's be able to assess this creature, look at its eyeballs, touch its flesh, feel the differences in texture and makeup of all the different parts of this creature. The blood vessels are actually red, and we can even make slides from tissue that's relatively fresh. So we're going to have a lot of intact cells to observe. So there's a lot of implications for using this design inference in the biology classroom. This is just a start on that idea. I hope it's been educational and encouraging for you. If you'd like to find out more, please check out homeschoolscience.org, and please stay tuned for more Boyd biology bits as we get ready for our first year here, fall 2021. Thanks.